Hello, my name is Travis Monk. This is one part of a series of videos involving cell biology. This video will provide an overview of some of the different ways that materials can move into and out of cells. The picture on the bottom of this slide illustrates the two main types of transport that will be described in this video: passive and active transport. The first type of transport that will be elaborated on in this video is passive transport. The term passive transport describes the process quite well. It's a means of transport, or a way that something can move in and out of a cell, and it's passive, meaning that it occurs naturally, without being acted on by outside forces. There are a few important characteristics of passive transport. First, the process requires no input of energy. Second, there is a movement from high to low concentration. Materials spread out, so to speak. The illustration on this slide exhibits what you would expect to have happen in passive transport. This picture shows that the vanilla smell, represented by green spheres, moved from an area of high concentration within the bottle to an area of low concentration with the rest of the room. There are many different types of passive transport. You may be a bit familiar with the first two types that are exhibited here: diffusion and osmosis. Simple diffusion is a type of passive transport where there is a movement of materials across the cell membrane without help. In the picture to the right, for example, there is a high concentration of some substance outside of the cell. Over time, the material moves through the cell membrane itself until the concentration eventually equalizes. Osmosis is simply a type of diffusion that involves water specifically. You can't sleep on your book and learn anything for a number of reasons. Even if you could, this wouldn't be learning through osmosis, as it has nothing to do with the movement of water. Facilitated diffusion is another type of passive transport. The term facilitate means to make an action or a process easier. One definition of a teacher is a facilitator of knowledge. With this type of passive transport, proteins within the cell membrane facilitate or help with the movement of stuff across the cell membrane. The reason that it is necessary is because some materials, particularly large molecules or ions for example, have trouble moving through the cell membrane itself. There are two common means of facilitated diffusion, which are ion channels and carrier proteins, both of which are exhibited in the picture to the right. Ion channels, that are shown on the top, provide a pathway for ions or charged molecules to travel through. Carrier proteins, exhibited on the bottom, change shape to allow particles, often especially large ones, to move into or out of the cell with ease. Facilitated diffusion is a type of passive transport for two reasons. First, no energy is required for these processes to occur, and second, there is a movement of something from high concentration to low concentration. There are three different types of environments that cells can be in at any given time. These three different types of solutions will be described on the next slides. There's a bit of background information, however, that will be important before describing them. Every solution that a cell can be found in, and every cell itself, is made up of water, a solvent, and a wide variety of other stuff, the solute. The percent of water and the percent of that stuff, or the solute, inside and the outside of the cell can vary considerably. What will happen in every circumstance is that the water and the stuff inside and outside of the cell will try to move from high to low concentration because of diffusion. The picture on the right exhibits the net, or the overall flow of materials, from the left to the right-hand side. There was more starting material on the left than the right, and things spread out until things evened out, or they reached equilibrium. The first of the three different types of environment that cells can be in is referred to as a hypertonic solution. The picture on this slide both shows what happens in this type of solution and explains what occurs at the bottom. The prefix hyper means in excess or too much. Someone who is hyperactive, for example, is excessively hyper. The hyper in hypertonic refers to the amount of stuff, or the amount of solute, that's on the outside of the cell compared to the amount of stuff or solute that's on the inside of the cell. The pink spheres in this picture represent the solute, and there is clearly a higher percentage outside of the cell than within the cell. The small purple or blue spheres represent water. Since the percent solute is higher outside the cell, the percent water is lower outside the cell. As this picture illustrates, most solute molecules are larger than water molecules. In fact, some solute is so large that it can't pass through the cell membrane on the cell because it's selectively permeable at all. Water, on the other hand, can move rather easily into and out of the cell. What happens in a hypertonic solution is that the higher percentage of water inside of the cell moves out in an attempt to equalize the percentages of stuff and water inside and outside of the cell. As a result, cells shrink in size. The second type of solution, again exhibited and described in this picture, is a hypotonic solution. The prefix hypo means the exact opposite of hyper. If someone is hypoglycemic, that means that they have low blood sugar. The hypo in hypotonic refers to the fact that there is less solute on the outside of the cell than there is on the inside. Hypotonic solutions have a higher percentage of water on the outside of the cell than the inside, and the same principles hold true as we described in hypertonic solutions. Water will move from high concentration to low concentration. The end result, as illustrated in the picture, is that water will move into the cell, causing it to swell in size. Isotonic solutions, as the picture shows and the descriptions explain, have equal amounts of stuff and water on the inside and the outside of the cell. These amounts are balanced, so the cell can be said to be in equilibrium. The prefix iso means the same. The terms isotope and isomer that we described earlier in the year all had the same something, either the number of protons or the chemical formula, in my examples. Since cells in this type environment are in equilibrium, there is the same amount of stuff and the same amount of water inside and outside of the cell. There is no net movement of water. Diffusion of materials into and out of the cell occurs at the same speed. Cells need lots of nutrients, and there are a few nutrients just floating around in the environment. Cells need lots of water, but there may not be a lot of water available in the environment at some times. Cells can't always just rely on the natural movement of materials, or passive transport, for survival. Sometimes what cells need is to expend energy in order to get what they need for survival, working against the concentration gradient. The picture on the right exhibits how, sometimes, cells need to expend energy to get more of something against the flow, so to speak. In the previous slide, I described how sometimes cells need to expend energy to get things to move against the flow. This type of transport is referred to as active transport. In active transport, the cell expends energy to move materials from a low concentration to a high concentration. The picture on the right shows the types of passive transport we discussed earlier on the left, as well as the new type of transport, active transport, on the right. The energy needed to fuel this process is provided by the molecule boxed in green, ATP. ATP, or adenosine triphosphate, is the energy molecule used to fuel all cell processes. This high-energy molecule is produced by mitochondria, an organelle within the cell, which we described last unit. ATP will be described in more detail later. Just like there are many types of passive transport, there are numerous forms of active transport. One of the main classifications of active transport is called endocytosis. The prefix endo means to go into, and cyto refers to the cytoplasm. Putting the two words together, this is the type of active transport by which materials are moved into the cell. There are two different forms of endocytosis, which are pinocytosis and phagocytosis. Pinocytosis is a form of endocytosis by which materials that are dissolved in water are moved into the cell, while phagocytosis is the means by which cells take in materials that are so large that they couldn't otherwise get into the cell. The process of phagocytosis is exhibited on the left. You can see a large particle that the cell membrane wraps around to form a vesicle, which is then incorporated into the cytoplasm. White blood cells perform phagocytosis on invasive bacteria and viruses to prevent them from causing further harm to the body. Pinocytosis, as shown on the right, works on the same premises. Dissolved materials are wrapped up by the cell membrane, forming a vesicle, which can then enter the cell. Materials can leave the cell in much the same manner in which they enter the cell through a process called exocytosis. The prefix exo means to leave, and cyto again refers to the cytoplasm, so the naming process makes some sense. This process is endocytosis backwards, so a vesicle, usually produced by the Golgi apparatus, comes in contact with the cell membrane. When it does so, the outside membrane of the vesicle incorporates itself into the cell membrane and releases materials into the environment. One of the functions of this process is to remove waste from cells. In addition to the removal of waste, many substances such as hormones are packaged and exported so that they can be used by other cells of the body. The release of neurotransmitters, or chemical messengers in the brain, using this process allows neurons, or brain cells, to communicate with one another. That is the end of this video summarizing different forms of cell transport. If you're interested in learning about any other topics involving cell biology or any other themes of biology, please subscribe to my channel. Thank you.